Welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you so much for joining us for another segment. We'll be speaking with Dr. Paul Lammers in this segment. He's joining us here as President and CEO at Triumvira Immunologics to talk about the company's work in the development of some novel T-cell therapies to treat cancer. Welcome to Health Professional Radio, Dr. Paul Lammers. Thank you for joining us this morning.
Thanks, Neil. Great to be here.
Well, tell us a bit about yourself and what led you to Triumvira.
I'm a physician and biologist by training, and I've been in drug development basically my whole career, 34 years in pharma and biotech as a drug developer, got involved in quite a few drug approvals over the years in different therapeutic areas. However, the previous company and this company, I've been involved in cancer, and what is better than to wake up in the morning and jump out of bed to go to work and trying to find a cure for cancer, right? I mean, we're all faced with that in our family, circle of friends, people that we know. So I was really excited. You know, I went to Toronto in 2017 to meet the co-founders of Triumvira, Jonathan Bramson, a professor of immunology at McMaster University in Hamilton, Ontario, out of whose lab the technology came forth, and Brian Bloom, the CEO of Bloom Burton, an investment advisory firm out of Toronto, and they're involved in a lot of Canadian biotech startups. And I was very intrigued by the technology, T-cell antigen coupler technology, especially because it's so well differentiated from CAR-Ts. And I thought it would be great to build a team and a company around it. So I joined January 2018 as the new president and CEO. At that time, there were two scientists in Jonathan Bramson's lab who were employees. I was employee number 3. Right now, we have 75 dedicated professionals in the company. We are about 60% U.S. and 40% Canada. Our research and process development team is up in Hamilton, Ontario, in a beautiful lab facility outside of McMaster University. Our headquarters is in Austin, Texas, where I'm located. Then we have also a GMP manufacturing suite in South San Francisco. And we have people basically from all the way from Seattle to Florida and Boston to San Diego. So we started social distancing before it became an issue.
If you would, tell us a bit about the current treatment landscape for solid tumors, and why does it remain a medical unmet need still?
Well, clearly there is a whole plethora of approved therapies for different types of solid tumors. And it starts with surgery, then there's radiation, then there's the well-known and feared chemotherapy that is still being used. If you think about it, more than 50% of all cancer patients in the world are still being treated with chemotherapy. And then there's all kinds of monoclonal antibodies, more targeted therapies, including cell therapy that is being used. Of course, cell therapy has not been approved in the solid tumor space. Cell therapies, especially CAR-Ts, have done phenomenally well in heme malignancies like lymphoma, leukemias, and multiple myeloma. But solid tumors is a big hurdle for CAR-Ts, and we can talk a bit about why that is. So truly, we feel that as an engineered T-cell therapy company focused on solid tumors, there is a huge unmet medical need still in various solid tumors. Obviously, we've made a tremendous progress in the treatment of different cancers, but certain cancers are still very difficult. If you think about, skin cancer like melanoma or pancreatic cancer, some of them are, or brain cancer, for that matter. Some of them are really, really difficult. But even, lung, colon, gastric cancers have a very high unmet need. And a lot of patients break through on different therapies and ultimately succumb to the disease, which is, horrifying.
Well, what makes some of those that you did discuss as being hard to treat? What makes them hard to treat? Is it something across the board in each of those types of tumors, solid tumors, or do each of them have some subtle nuance that poses such a challenge to cause them to be limited? And based on those limitations and the current T-cell therapies advances that you mentioned, what is Triumvira doing at this time in their development process?
Well, the first challenge is cancers are really smart, which means is if you think that a certain, pathway within the tumor that leads to the tumor and leads to the growth of the tumor, if you block that through some kind of, product that you're administering to the patient, it may work for a while, but what the tumor does, they mutate around the block. And which means is that the number of mutations in tumors can be overwhelming. So you think you're treating a certain type of mutation, for instance, HER2 positivity or EGFR or go down the list of various things. And before you know it, the cancer comes back and the tumor has changed in terms of its genomic makeup. So the mutation, as they call it, in tumors can be really high, and that makes it really difficult to stop the tumor from growing. Also, especially in what you call undifferentiated tumors. So tumors have different types. When you look at the pathology, you have variable differentiated, which means they look very much like the normal tissue where they originate from, to very undifferentiated, which are like, a bunch of, angry-looking cells under the microscope that proliferating uncontrollably. And some of these tumors grow really, really fast and to the detriment of the patient, clearly. Cell therapy is unique because people say that the T cell is the most elegant drug ever developed for cancer. Because what is better, Neil, to try to use a patient's own immune system to try to kill their own individual tumor cells. And that's something that we're trying to do. And obviously, as I said, it has worked well with CAR-Ts for heme malignancies. But the solid tumors is a challenge. We feel that our TAC T cells, as they're called, have a unique structure and biology that makes them ideal for this kind of what you call adoptive cell therapy or T cell therapy. So what we do is just a process. So we take blood from a patient, Neil. We send it to the lab. We extract the white blood cells, which are critical for your immune system. And there are two types of white blood cells, T cells and B cells, or T lymphocytes and B lymphocytes. We focus on the T lymphocytes. We genetically engineer them to express a vector on the surface of these T cells, and that structure is known as the TAC, or T cell antigen coupler. Then we expand those TAC T cells in the lab until we have billions. And then we basically harvest the cells, we freeze them in, we ship them back to the clinic, and the patients get his or her own cells reinfused, but now they aren't with this vector on the surface that recognizes and kills their own individual cancer cells. I mean, there's like the ultimate personalized medicine here.
Absolutely.
You can think about it. So, and that's how CAR-T was developed. And as I said, a lot of success against lymphoma, leukemias, and multiple myeloma, but in the solid tumor space, because if you think about it, in the heme space, lymphomas, leukemias, and multiple myeloma are diseases of bone marrow or your lymphatic system or your blood system. So it's relatively easy for cells to get there. Solid tumors is different. So the cells, you inject them into your intravenous, they circle around, right, to your circular system. They have to get out of the bloodstream, so extravasate, then go to a stroma into an organ and try to find that tumor cell. So you really need to have very fit cells because it's a long way to get there, so to say. A lot of hurdles that these cells have to overcome to finally get to that tumor cell and then try to kill it. And therefore, you need to have fit T cells. And that's why our biggest differentiator with our TAC T cells compared to CAR T cells is CAR T cells are built to produce cytokines to kill tumor cells. That's great, but there are two problems with it. One is when you manufacture CAR T cells, they're already in that process of manufacturing. They start producing cytokines because that's how they're built, which means is these poor cells have to run a marathon. They exhaust quickly, so they don't have the stamina to stick around and try to find that process of finding that solid tumor cell. Secondly, because they overproduce cytokines way more than you need to kill a tumor cell, you lead to severe toxicities. And that's still one of the biggest drawbacks of CAR T therapy is the toxicity associated with it. That could be either what you call CRS or cytokine release syndrome. It could be a neurotoxicity phenomenon, which is also devastating to the patient, also to the patient's family, by the way. So we feel that our TAC T cells don't, they have a very, as I said, the biology and the structure of a TAC T cell is totally different than a CAR T cell, and it leads to, the fact that the TAC T cells only truly get activated when they're meeting up with a cancer cell in that tissue. We are running our first phase 1/2 clinical trial targeting HER2, a well-known target in solid tumors. And we also will bringing in our next exciting target is a TAC T-cell directing against Claudin 18.2 as another target in solid tumors, especially gastric and pancreatic, into the clinic later this year. Our website is www.triumvira.com. We're an exciting, engineered, different and new cell therapy company, and we hope we can have major success in the solid tumor space.
Well, Doctor, I appreciate you taking time with us this morning here on Health Professional Radio. Hopefully you'll return and give us some updates as the clinical trials that are planned go forward, okay?
Absolutely, Neil. Appreciate it. Thanks so much.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, and Dr. Paul Lammers. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at Anchor, Spotify, and be sure and subscribe to our YouTube channel at youtube.com/Health Professional Radio.